Welcome to an introduction to pharmaceutical microbiology. Today we're going to be talking about bacteria and their classification. Bacteria do not only cause disease, but they are responsible for producing life-saving antibiotics. In pharmaceutical microbiology, we study microorganisms that affect drugs and human health. Pharmaceutical microbiology examines harmful microbes that causes diseases or contaminate medicines and are useful to produce antibiotics, vaccines, and biotech products. This field ensures drug quality and safety through sterilization, disinfection, and microbiological testing. Now, looking at the structure of bacteria, we can see it has some important organelles like the cell wall, which provides shape and protection, the cell membrane, which controls what goes in and out of the cell, the cytoplasm, where all metabolic reaction happens, the ribosomes for protein synthesis, production of proteins, the nucleode, which is the region containing the DNA. Since they don't have nucleus, remember they are prokaryotic organism, they do not have a nucleus. The plasmids, which are extracellular DNA pieces that are capable of reproducing independently apart from the DNA, the main DNA. We have the flagella and the pili used for movement and attachment, as you can see on the surface of the bacteria. These are just some of the basic organelles that is contained in a functional bacteria. An important thing to note is the classification of bacteria. We classify bacteria based on the shapes. We have those that are the spheres, those that are the rods, those that are spiral. For the spheres, we also call the spheres cocci. For the rods, we also call the rods bacilli. And we have the spirals. The spherical bacteria, which are the cocci, the plural of coccus, can be further classified into diplococci. Those are the spherical bacteria that comes in a pair. An example is Streptococcus pneumoniae. We have the streptococci, those that form a chain-like structure or like a strand. An example is Streptococcus pyogenes. We have the tetrad, those that form a network or a connection of four. We have the staphylococci, those that forms clusters. An example is Staphylococcus aureus. We have the Sakina, or you can see Sakini, those that form a cubic form or a cubic shape. An example is Sakina ventriculi. Moving over to the rods, which are the bacilli, the rod-shaped bacteria, bacilli, plural for bacillus. It is further classified or further divided into the chains of bacilli, which an example is Bacillus anthracis, that is the bacteria responsible for anthrax. We have the flagellated rods, an example is the Salmonella typhi. We have the spore formers or spore containing or spore forming bacilli. An example is Clostridium botulinum. That is a bacteria that is that acts as a neuromuscular blocking agent. Moving over to the spiral, we have the vibrous. The spiral are spiral-like bacteria. They are bacteria that you can say they are in a comma shape. Um, they are well twisted. We have the vibrous. An example of vibrous is the Vibrio cholerae. That is the bacteria responsible for cholera. We have the spirilla. Yes, spirilla, which is the Helicobacter pylori, the bacteria responsible for ulcer. And we have the spirochetes. Don't mind my pronunciation, guys. The spirochetes. An example is the Treponema pallidum. And these are all bacteria or classification of bacteria based on shape. Based on shape. Moving over to the next classification of bacteria, we can classify bacteria based on the cell wall staining. Now, bacteria are made up of cell wall, which of course give the bacteria its shape, whether the bacteria would be a spherical shape, a rod, or a spiral shape. This cell wall contains cell membrane, and the cell membrane contains special molecules called peptidoglycan. So, bacterial or classification of bacteria based on cell wall staining We have the gram positive and we have the gram negative. The gram-positive bacteria have a thick peptidoglycan walls. That means they have a thick layer of peptidoglycan and they stain purple. An example of this bacteria under this class is Staphylococcus aureus and Streptococcus pneumoniae. We have the gram-negative bacteria that has a thin peptidoglycan wall or peptidoglycan layer plus an outer membrane. That means the peptidoglycan layer is bound by an outer plasma membrane and an inner plasma membrane. And of course, it stains pink or red. An example is the Enterobacter coli, which is E. coli and Salmonella, or you can see Salmonella typhi. Now, this is the classification of bacteria based on cell wall staining, gram-positive bacteria and gram-negative bacteria. The gram staining procedure, the gram staining technique involves four basic steps. The first step is adding a particular dye, which is crystal violet, to stain the specimen. The second step is adding iodine, which is a mordant that makes the dye less soluble so it adheres to the bacterial cell wall. The third step is adding of alcohol to decolorize or wash away stain from gram-negative cell wall. And the fourth step is adding a counter stain, which is safranin, that allows the dye adhere to the negative cell wall. And don't forget that the final stage of the gram staining procedure technique, which is the gram positive bacteria staining in purple and the gram negative bacteria staining red. Another classification of bacteria is based on their oxygen requirements. We basically have the aerobes and the anaerobes. Aerobes are those that need oxygen to survive, and anaerobes are those that can live without oxygen or in the presence of low oxygen. We have the obligated aerobes, which require oxygen and can't survive without it. We have the facultative aerobes, which uses oxygen but can survive without oxygen. We have the aerotolerant anaerobes, which do not use oxygen but can grow in its presence. We have the obligated anaerobes, which is unable to use oxygen and can't survive if it is present. And we have the microaerophy, which uses oxygen but at a very low concentration. Another classification of bacteria is based on their temperature preference. We have those that can grow and survive at extremely high temperature. They are the extreme thermophiles. Those that can grow and survive at high temperatures, they are the thermophiles. Those that can grow and survive at moderate or optimum temperature, they are the mesophiles. And those that can grow and survive that they require a very low temperature, they are the psychrophiles. So this is the classification of bacteria based on their temperature preference. We also classify bacteria based on their pathogenicity. We have the non-pathogenic bacteria, which are harmless and they are sometimes beneficial. These are bacteria that are normal to our physiological systems. They are the bacteria that are in the microflora or in the natural microbiomes of the organs of the body, especially in the GIT. We have the opportunistic pathogens, which are harmless but can cause disease when the immunity is low. If for example, someone is diagnosed with HIV, this bacteria can actually cause serious infection. An example is the Pseudomonas aeruginosa. Don't mind my pronunciation, guys. Then we have the true pathogens. Those are bacteria that always causes disease. An example is the Mycobacterium tuberculosis that is responsible for tuberculosis. They always cause disease. Finally, we are going to be looking at the prokaryotic cells versus the eukaryotic cells. And of course, bacteria are prokaryotic in nature, and we have the unicellular eukaryotic cells. Of course, they have similarities. They both contain DNA, they both contain ribosomes, they both have cell membrane, they both contain cytoplasm, but they have differences. Like for bacteria or prokaryotes, they have simple structure, eukaryotes have complex structure, prokaryotes are of small size, eukaryotes are of large size, and you can pause this video to look at this Venn diagram of the difference and the similarities between prokaryotes and eukaryotes. Now, of course, bacteria divide by simple asexual method, which is called binary fission. The bacterial cell copies its DNAs, elongates, and then splits into two identical daughter cells. This is how bacteria reproduce by binary fission. If you know this video was helpful and you learned a lot, like and subscribe for more on David Charis Pharmacology. And if you want me to take any other topic, you can comment below.